So I would like to talk about angiogenic sprouting, and just to remind us all and why it's interesting and important to study angiogenesis. And we've heard about that already yesterday in a couple of very interesting presentations. Of course, the hope is that on the one hand, you can stimulate and improve regenerative processes, and on the other hand, there are various pathologies where you would like to inhibit angiogenesis and prevent, for instance, the development of eye diseases or in cancer growth and metastasis. The model system I will be talking about is the retina of the mouse, and in the mouse, the vascularization of the retina occurs after birth, so not in the uterus, so that's a great advantage. And what you can see here at different postnatal stages, the vascularization starts here in the center of the retina. That's also where the optic nerve enters, and then progressively extends towards the periphery. And at P7, you've almost reached the periphery, and you have initially a single layer of blood vessels, which is called the superficial layer. And then at these stages here, there is sprouting into the deeper retina, and two additional layers are established. And these are advantages really of the retinal system, that there is a clear spatial temporal development here. And if you look at the earlier stages, so before P7, you have only a single layer of blood vessels that can be very nicely analyzed by whole mount imaging. So you see the whole vascular bed with all side branches and all aspects. So that's a great advantage. And that's really how an image looks like here. The different cell types that are relevant have been stained: the endothelial cells, vascular smooth muscle cells in red here, and the pericytes. And what is also nicely exemplified by this image is that if you look here in the central retina, these are the blood vessels that have formed first, and they are the most mature ones. So you can here clearly identify the arteries, two of them that are already covered by vascular smooth muscle cells. The vein here is not yet covered by vascular smooth muscle cells, and you have also capillaries. And of course, you can look now at the interactions between the different cell types, for instance, the interactions between pericytes here in green and the endothelial cells. In the periphery, you have the most recently formed vessel network, and you have something that resembles very much a primitive capillary plexus, so not very highly organized vessel network. You have still some avascular tissue in the periphery, and you can see here and appreciate the endothelial sprouting that extends the vascular network. Let's have a closer look at the endothelial sprouting. So this is now with a fixation condition that is optimal for preserving and visualizing the filopodia that are extending from these sprouts. And here you see in the scheme that there are different cell types that are contributing to the sprout, and the tip cells that are leading and extending this filopodia, and the stalk cells that form the base of the sprout. All this is very dynamic in various ways. So if you look, for instance, here in the zebrafish intersemitic vessel, you can see the extension, the dynamic extension of filopodia. I hope you can see that also in the back of filopodia that are stretched out, and they are pulled back in a highly dynamic fashion. And also the tip and stalk cells are shuffling and competing for the tip cell position. So these are by no means fixed fates, but there's actually a very dynamic switching of these positions. On the molecular level, there are attractive as well as repulsive cues that control this process. The most important attractive cues belong to the vascular endothelial growth factor family, and we have the VEGF gradient that is expressed in VEGF-A is expressed predominantly in avascular tissue. We have also other sources of growth factors like macrophages that produce, for instance, VEGF-C, and the corresponding receptors, VEGF receptor 2 and VEGF receptor 3, are expressed by the endothelial cells. And there's also evidence from Lena Claesen-Welch and other people now that there are 2 or 3 heterodimers, and it will be interesting to figure out what they are doing in comparison to the homodimers. So sprouting alone is of course not enough to make new blood vessels. You need other processes, and that's shown in this cartoon here. So sprouts need to make new connections, and these connections have to be then converted into blood-carrying tubes, and these are actually steps that are still relatively poorly understood and that need to be investigated much more carefully in the future. Another important aspect, of course, of the angiogenic growth is endothelial cell proliferation. You need more endothelial cells to make more extensive vessel networks. So important question now is, what is controlling these different behaviors of endothelial cells in growing vascular beds? If all endothelial cells would try to behave like tip cells, that wouldn't be good. You would be destroying existing vessels, and you would lose perfusion. On the other hand, if there are not enough tip cells, you don't get enough growth and sprouting. And work really from many groups and over quite a couple of years has developed this model here. So tip cells express high levels of a notch ligand called delta-like 4, DLL4, shown here. And that leads to the activation of notch receptors in the adjacent stalk cells, and it's thought that this represses then the expression of VEGFR2 and VEGFR3 in the stalk cells, so that these cells have lower levels of these receptors and respond less to tissue-derived growth factors, VEGF-A and VEGF-C. So that's the model. And it's actually not only a concept that applies to vascular growth. Here's a picture from a review that I wrote a couple of years ago. Also in the Drosophila system, for instance, in the developing tracheal system, it's thought that there's a similar principle of notch ligand expressing tip cells that signal to adjacent cells and suppress certain behaviors. So that seems to be a general concept that applies to network formation processes in different species and tissues. I'm sure you're all familiar with the notch pathway, just the key principles. So we have membrane-anchored ligands belonging either to the delta family, like delta-like 4, or the jagged serrate family, and I will be talking about a ligand JAGED1 briefly. And these membrane-anchored ligands interact with membrane-anchored notch receptors, and the binding leads to cleavage processes that release the extracellular domain of the notch receptor, and also release the intracellular domain shown here, that then is translocated to the nucleus and interacts with a couple of other factors to control gene transcription. And there's a whole large range of different target genes that are important. Many of these target genes are, again, transcriptional regulators, so you really have a quite complex and powerful transcriptional cascade. And also, what is also important is that there's a positive feedback loop. So one of the targets of notch signaling is actually delta-like 4 expression, which is upregulated and thereby enhancing notch signaling because there are new delta-like 4 notch interactions. And of course, this also imposes some questions. So if I have a signaling loop like this one shown here, why is it then that the whole system doesn't come to a standstill? Because notch signaling will lead to delta-like 4 expression. Delta-like 4 expression will lead to further notch activation, and that would just stop this whole process. And there are, of course, many answers to this. I'm not going to talk about the regulation of the stability of notch today, but we are investigating the role of the E3 ubiquitin ligase FBW7 that is degrading active notch and thereby limiting the length of the notch signal. And I will briefly talk about data that we've published on JAGED1, which actually is an antagonist of delta-like 4 notch signaling in this system and prevents overactivation of the notch pathway. And this depends on glycosylation steps that I just really briefly mentioned now that modify and add notch receptors and add certain sugar chains that are very important. So if you look now at the expression of the notch ligands delta-like 4 here in red and JAGED1 in the growing vasculature, you can appreciate that delta-like 4 is delta-like 4 protein is decorating the sprouts, and jagged is actually present, but only at a very low level in the sprouts, and it's more found in the stalk cells and in the capillaries. If you look here at higher magnification, you see two tip cells with high levels of delta-like 4. There's also some delta-like 4 in adjacent stalk cells, but these stalk cells have high levels of JAG1. So what is JAG1 doing? And initially, we were sort of expecting that it's known that it can activate Notch receptors, and we were expecting that it's actually having a similar role as delta-like 4, but actually it turned out it's doing exactly the opposite. So when we used the tetracycline system, as introduced by Ralf H yesterday, to overexpress JAG1 here in endothelial cells, what you get is enhanced vessel growth, so there are more branch points and there's a larger area covered by endothelial cells. And this bottom picture just shows you the level of JAG1 overexpression in the endothelium. If you do the complementary experiment and use the Grelox-P system to delete the JAG1 gene in endothelial cells only, and that's only these are all postnatal manipulations. If you would do this in the embryo, this would lead to embryonic lethality. So postnatal deletion of JAG1 in endothelial cells leads to a smaller number of branch points and a reduction in the number of the endothelial cell covered area, as you can appreciate here. And when you look at the angiogenic front, at the sprouts, I think even without quantitation, you can immediately appreciate that there are fewer tip cells and fewer filopodia. We've done the quantitation, so this is shown here. So all this is really showing that Jagged is promoting angiogenesis, which is the opposite of what delta-like 4 is doing. And to really prove that Jagged is doing this by modulating delta-like 4 notch signaling, we did a couple of experiments. So one thing was to look at notch targets, and that's shown here. So one important notch target is this transcriptional regulator HEY1. In control retinal vessels, HEY1 expression is very, very low. So you're hard pressed to find some endothelial expression. However, in the Jagged loss of function mutant, again, that's just an endothelial cell specific mutant, HEY1 expression is strongly upregulated in the vessel beds, as you can appreciate here. And we also did then some rescue experiments. So what happens now in this Jagged loss of function mutants when you block notch signaling? So we would release this blocked angiogenesis, and we would expect sprouting, and that's indeed the case. So here you have again a control retina and a Jagged loss of function retina, and you can appreciate the differences in vascularization. If we treat the control retina with an inhibitor of gamma secretase, which is an important enzyme controlling the cleavage and the activation of notch receptors, this leads to enhanced sprouting and proliferation here in this region. At the peripheral retina, that's actually the region that has grown during this time window where we administered the inhibitor. And we do the same experiment now in a Jagged loss of function mutant. You can see that we also get enhanced sprouting and proliferation again here in the distal region, whereas the density of the vessels that have grown before the DAPT administration is still reduced compared to the control. So that showed really it's a problem, the enhanced notch signaling in the Jagged loss of function mutant that inhibits angiogenesis. And we also then did some experiments, and I shall show you one example to prove that Jagged1 and delta-like 4 are indeed competing, that it's a trans effect and not some other model that is relevant. And what we did here was to code immobilize delta-like 4, recombinant delta-like 4 or Jagged1 proteins on dishes and place endothelial cells on top of that. And we then looked by qPCR at targets, no notch targets that are shown here. So it's again HEY1, HES1, and delta-like 4. And we looked at the upregulation of these targets. And delta-like 4 leads to clear upregulation of all three targets. Jagged1 is less potent in this assay. That's also not surprising, but it leads also to the upregulation of, for instance, delta-like 4 in HEY1 expression. when we engineered endothelial cells that overexpress one of these fringe family glycosyl transferases that are modifying the Notch receptor, what happens is that actually delta-like 4 induced responses are strongly enhanced. And you can appreciate that here it's most obvious for delta-like 4, but also there's a very clear effect for HEY1. However, jagged responses are even further reduced compared to the control, and you get a very small increase. And now what we did here in this experiment was to take the very same amounts of delta-like 4 and jagged 1 that we used in the individual experiments and combine them 1-to-1. And what you could expect is that there's maybe just an additive effect, but instead, actually, what happens is that there's competition of delta-like 4 and jagged 1, and you get an intermediate response. So jagged 1 in this system can compete with delta-like 4 Notch interactions. So this led us really to this model, and actually shortly afterwards, the group of Klaus Kästner has also shown in pancreas development that jagged is acting as an antagonist of delta Notch signaling in this system. So it seems to be a general principle that could apply to other tissue systems as well. So this model, as you will see here, still strongly relies on the link between Notch and VEGF receptors, and that was the next thing that we investigated. So is it really this link that is responsible for the different behaviors of tip and stalk cells? And we did a couple of genetic experiments again, and what I'm showing you here is again a control retina at postnatal day 6. And here, in this case, we induced endothelial cell-specific delta-like 4 mutants. So this is again a postnatal gene targeting experiment. And what happens is, and you can appreciate that easily, is this tremendous overgrowth and over-sprouting of endothelial cells, which leads to a very dense network. There are lots of filopodia extending tip cells. So that is sort of not surprising. That's the expected effect. We also generated VEGF receptor 2 loss of function mutants that are shown here. And again, actually, the expected thing happens. We get strongly reduced vascularization of the retina. It's a very sparse network. If you look at higher magnification, there are very few filopodia extending cells. So clearly, that was also what we expected. And the question was now, what happens if we combine those two mutations? And if Notch is really upstream of VEGF receptor 2, it shouldn't matter anymore. If you don't have VEGF receptor 2, you cannot get angiogenic growth. But very much to our surprise, if we generate VEGF receptor 2 and delta-like 4 double mutants, and we also validated that really both genes were effectively targeted in the endothelium, you get strong angiogenic growth even in the absence of VEGF receptor 2. And that was really a big surprise, clearly not what we had expected. And that's the quantitation of this data. So we also then used other approaches to validate the same point, and here's another Notch pathway mutant that I want to introduce. It's an RBPJ mutant. It's an important transcriptional regulator that is essential for Notch signal transduction. And again, when we target RBPJ in the postnatal retinal and postnatal endothelium, here what you see is endothelial cell overproliferation, a very dense network sprouting, and also reduced outgrowth towards the periphery, which is also typical for delta and other mutants. So what we did was then to administer anti-VEGF blocking antibodies for 3 consecutive days to mice. And in control mice, you can see that you can effectively reduce angiogenic growth with this anti-VEGF-A blocking antibody. So the density and the sprouting is strongly reduced. However, in the RBPJ mutants, there was barely any effect. We did the same with anti-VEGF receptor 2 blocking antibodies that you can see here. And again, there's a clear effect in the control retina, but not in this Notch pathway mutants, which is consistent with the genetic experiment I showed you previously. And this is just at higher magnification to show you that here the anti-VEGF-A treatment is really effective. If you look at higher magnification at tips, for instance, you can see, first of all, there are very few tip cells, and filopodia are largely reduced, very short or absent. And this is not the case in the RBPJ mutant treated with the very same anti-VEGF-A antibody. So what is going on? And one question was, is VEGF receptor 2 now not so important really for Notch-mediated angiogenesis? And we looked here by Western blots from lung lysates obtained from these mice. We used lung lysates because lungs have a large content of endothelial cells, about 20%. If you would use retina, it's impossible. Only 1 in 2,000 cells roughly in the retina is an endothelial cell, so we couldn't take a whole tissue lysates for biochemistry. But for the lung, it's possible. And what you can appreciate if you look at VEGF receptor 2 levels, there is a small increase of VEGF receptor 2 levels in the ABPJ mutant lung. However, it's comparable to the increase in V-cadherin, and also by looking at stainings, we think it simply reflects the increase in the number of endothelial cells in those lungs. So it's not truly a strong upregulation of VEGF receptor 2. Various studies from Karya Litalo and Nathan Lawson and other groups have shown that also VEGF receptor 3 is regulated by Notch. And when we look at VEGF receptor 3 and compare the levels here in the control and the ABPJ mutant, there is indeed a very dramatic upregulation of this receptor. And it's not just the upregulation of VEGF receptor 3, it's also the activation of VEGF receptor 3 that is strongly enhanced. And what we did here was a phosphotyrosine IP experiment where we looked then at the levels of VEGF receptor 3 that are pulled down, and what you can see is that there is a very strong activation in ABPJ mutant lungs compared to control lungs. For VEGF receptor 2, again, we see a slight increase, but this we mostly attribute to the increase in the number of endothelial cells. We also looked at VEGF receptor 1, actually, and VEGF receptor 1 levels go down, and also the tyrosine phosphorylation of VEGF receptor 1 are reduced. So this is arguing against the potential role of VEGF receptor 1 in sort of compensating for the absence of R2. And we also checked the same in delta-like 4 VEGF receptor 2 double mutants. I didn't emphasize this so much, but VEGF receptor 2 is positively regulating VEGF receptor 3 expression, not just negatively regulating VEGF receptor 3 expression. So if you take both away, we still have a very robust upregulation of VEGF receptor 3 compared to controls. A bit less than in the delta-like 4 mutant, but you can still appreciate if you compare those two bands here, there is still a strong difference. So then we did some functional experiments, and because we didn't have the possibility to target yet more genes, we used chemical inhibitors here, and to test the role of VEGF receptor 3 kinase activity in this system. I'm not going to talk you through all these panels, I just show you the quantitation data, and we can start here, focus here with the sprouting data at the top. So if you have a control retina and you treat it with DAPT, the gamma secretase inhibitor, there's an increase in sprouting. That's not surprising. In VEGF receptor 2 mutants, you start with a lower number of sprouts, but if you give DAPT, you still get a very robust increase. However, if you now take a VEGF receptor 3 mutant and you block Notch and VEGF receptor 3 kinase activity with a kinase inhibitor called MAS51, which was previously published by Jonathan Sleeman, this increase is absent. And the same also applies to filopodia formation and vascular density down here. So we then also used other reagents to show that actually the combined inhibition of all VEGF receptors with this PAN blocking antibody, with this PAN inhibitor, it's not a blocking antibody, PAN inhibitor AL993 suppressed Notch-induced angiogenesis shown here. So and that's the quantitation of this data. So we come up with a modified model that has actually some important implications. So what we think is that this modulation of VEGF receptor 2 by Notch in vivo at least is weak or absent. And so this is not an important factor, and Notch is not modulating dynamically VEGF receptor 2 expression. Maybe there are other factors, but Notch at least would not lead to dynamic up or down regulation of VEGF receptor 2 in certain cells. What is very strongly modulated is VEGF receptor 3, and Notch can, of course, then give very dynamic patterns of VEGF receptor 3 up and down regulation. And when Notch is blocked, VEGF receptor 3 is strongly upregulated, as shown here, and is an important factor, probably not the only factor, but an important factor driving the angiogenic growth. And I didn't show you the data. This also cannot be blocked by adding ligand-binding blocking antibodies in vivo, suggesting that this might be ligand-independent, or as Kari Alitalo yesterday has suggested, maybe cis signaling, that perhaps the endothelial cells produce VEGF-C themselves, or it could be actually that just the sheer upregulation of VEGF receptor 3 to in such dramatic extent is enough to lead to activation of the kinase activity, which is also known for certain cancer cells or other cells that overexpress receptor tyrosine kinases. In these cases, you can also get ligand-independent activation. So this could be also very relevant for disease settings. So if you think about non-responders against anti-VEGF therapy in cancer or in that age-related macular degeneration, there is a certain fraction of patients that poorly or never respond to Lucentis, the anti-VEGF blocking antibody. This could be very important. In the future, it might be very important to determine the status of endothelial notch activation and predict the outcome of anti-VEGF therapy. So last but not least, I would like to thank the people who have collaborated to this work. I've highlighted the most important collaborators here: Georg Preyer, Antonio Duarte, Freddie Radke, and Bronislav Petowski at Int'l in New York. And in the lab, I'm very grateful to an extremely talented postdoc, Rui Benedito, who is now moving on to Spain this summer to start his own lab, and various other people in the lab that have worked with Rui. And I thank you for your attention. I'm happy to take questions.